is very important. Sometimes people will talk about bio-waste material. The moment you say bio-waste, most of the people will say, I don't want to use bio-waste as a material for my healthcare. Right? So that is why when I visited University of Cambridge, my mentor actually transformed the word. Instead of bio-waste, they said sustainable. Okay, same word, just changing. Okay, sustainable biomaterials, this is very important because as you all know, if I have to make a device, I have to go to Sigma or Ridge or any other companies. So as you see that product cost in mg few thousand. So what will be the final product cost? Right, so this is very, very important. We can work on fancy materials, make fancy devices, but ultimately when we have to actually utilize for the application, that is why you have to consider the cost. So that is why I thought, like if I am working in India, then definitely I have to choose the material which has to be sustainable, which has to be cost effective. That is very important. So that is why I thought that can we do some extraction procedure for the healthcare product development. So with this, I think I would like to go into, I always like to go into the stories, right? So because I think I would be thankful to Dr. Dr. Joshi, who is a rheology expert, and in fact, rheology is very close to my heart because we know that my lab, you will see that most of the processes we follow the liquid processing. So we start with the liquid, anything, whether it is solid, whether it is a semi-solid, doesn't matter. So we try to make it liquid, meaning either solution or suspension or emulsion, then we start fabricating things. So that is why very important to know the rheological parameters because ultimately your process control is the rheological control. If you have to repeat the same device with repeatability, that means we have to have the process parameter control, quality control. So that is why rheology is definitely an important thing. So in fact, throughout my research life, at least I can say that a lot of rheological studies we have done, and across the globe, whatever rheometers are available, all I have used. I started with TA instrument, Brookfield, Fungilab, Antampa now, recently, last couple of years, and of course, Malvern, and so on and so forth. So I think if I have to start a biomaterial, I think I have to talk about the history. That is more interesting, at least to understand how the field was evolved over time. So as you can see here, we get the inspiration from nature. If you are lazy and lying on the bed, then you can see the story of lizard, where you see that lizard has a fight, and ultimately you see the tail has cut down. And then eventually after a few days you see there is a growth of tail or whatever you say. So this is where we see that okay, lizard has a growth, so we can also have. Somebody chop off the hand or whatever ear or nose and it may grow over time. It doesn't happen. So that is why we need to go for regeneration. So here, interesting story is the Lindbergh, who was working in collaboration with surgeon Alexis Carrel. Right? They published a paper in 1930, actually 32, about the culture of organ. Right? So this was perhaps in the history, the first book where they are talking about organ culture. Similarly, they have done a very interesting experiment. So what they have done, they have used the perfusion pump to keep the organ alive for longer duration. Right? So that is why they are actually searching for a device or making a device where they can keep the organ alive for some time so that they can transplant it to other person. So based on the work, they got also Nobel Prize in 1930. So as you can see here, they written the book on organ culture, and also they developed a bioreactor for blood oxygenator and so on and so forth, or bioartificial livers. Similarly, so you can see there is another remarkable story where I think Brenmark was doing a very interesting experiment. He was actually looking at that how the blood flow can change the healing kinetics of the bone. So he devised a very narrow, fine titanium-based microscope. He wanted to put inside the bone to do the follow-up study how the titanium microscope can take the images over time, and then wanted to analyze how it is the blood flow can control the kinetics. So after the experiments, few weeks, he wanted to remove the implant or the microscope. He was not able to, because he found that actually microscope was fixed to the bone. In the history, for the first time, a man-made material which was grafted inside the bone was bonded to the natural material. So that is why he realized, okay, this is kind of eureka moment, because people were looking for different materials for the healthcare applications. So there are two schools of thoughts. One is absolutely inert material, and another is absolutely active material. So Lindbergh was looking at absolutely active material, and Halbert was looking at the inert material. And that is why Lindbergh has contributed in a way. He developed a microscope. It so happened that he chose the titanium as a material, and titanium was bonded to bone. So that is why he realized perhaps titanium can be used for bone bonding. So that is why he proposed a dental implant, and in fact till date, Denmark clinics is there, and they have a design set up. Still date, whatever proposed design he had, still in the market. And in fact, Denmark is the key person who has given the titanium as an implant, and today we know titanium implant is one of the best material for specifically for load bearing applications. So now another story is about Sir Harold Ridley. So he was a ophthalmic surgeon, and a patient came to him for eye-related problem. So then he asked the person that what is your profession? What is your profession? Then he said that, so I was a pilot in the Second World War time. So in fact in the First World War time, as you know, the canopy was covered with it was not covered. So pilots are exposed to the weather, like you can see that minus 40, 50 degree pilot is gliding the aeroplane. But in the Second World War time, so there was a canopy, and the canopy was made of PMMA, polymethyl methactylate. And he found that inside the eye there is a piece of foreign material. And also he found that even after so many years, it does not have any adverse effect. So that is where he realized that perhaps PMMA can be used for ophthalmic application. So that is where the definition of biocompatibility comes in. So if you are using a material for a application in a model for a certain period of time, so you cannot say that, okay, material was there in ophthalmic and did not have adverse reaction, therefore I can use it for heart or kidney. You cannot, right? So if it was used in the ophthalmic and it was used for 1 month, so you can say this material is biocompatible because I have done ISO 10993, I found all tests were clear, and it was used in ophthalmic application for 1 month in rabbit model. I can say this material is biocompatible in rabbit model for 1 month while it is implanted in ophthalmic location. This is very important. So this is what is the definition. Just by doing cell culture, you cannot say biocompatible. Cell culture, if you see cell is alive, then it is cytocompatible with this cell line for this many period or whatever, right? So, but interestingly, so when he found that, okay, PMMA was there, PMMA was inside the ophthalmic location, and it was right. So then he chose, okay, PMMA can be used for lens-related applications. In fact, as you all know, the cataract surgery, after cataract surgery, the lens will be removed, right? So after that, many people were blind. So he chose to use PMMA-based material that can be used as a lens. It can be placed in the intraocular lenses. So therefore, he is actually the pioneer for intraocular lenses, and later on it was several modification is done. So PMMA was made into PHEMA, so therefore it became a hydrogen-based intraocular lens. So there is another interesting story, as I mentioned to you, that there are two schools of thoughts. One is absolutely bioinert material, another is absolutely bioactive material. So when there is a bioactive material, there will be adequate host response, and when there is a bioinert material, there is anyone? So if there is a bioinertness, what will happen? Absolute inert is possible? Yes or no? Absolute inertness is not possible. So as you all know the PARL. PARL is the manifestation of the foreign body response. So how PARL is made? We take a piece of material, place inside the snail, and over, because of foreign body response, they start depositing. So exactly the same thing will happen. So though you decide to have a foreign inert material, but it will not be inert. There will be response, foreign body response, there will be neutrophil, macrophages, and then finally fibroblasts will come and they will try to wall off. What? So if my neighbor is not good, what I will do? I will make a thick wall of fibrous tissue. So fibroblasts will come and they will make a lot of collagen synthesis. Therefore, there is a pretty thick fibrous layer of collagen type 1 to wall off from the immune system. So that is what happens usually if I use a bioinert material. But there is another school of thought given by Larry Haynes. So he thought I can use the bioactive material. So in fact, he actually just did a PhD in glass from University of Florida. Then he was talking to a doctor who came back from Cambodia, Vietnam War. Then as the discussion happened, apparently he realized that doctor suggested it was very easy to save the life of the soldiers, but it was very difficult to save the limb of the soldiers. So then he was looking for a material which can save the limb of the soldiers. So but discussion happened, and after that he realized, because his PhD was on glass, so after discussion he realized that okay, we can use a little bit of calcium and phosphate and sodium along with the silica, so therefore we can make a glass. So arbitrary experiment, first experiment, and in 1959, he made a glass and he has placed in the rabbit model, and they are pulling the glass. Glass was not coming out because again it is in the history. First man-made material was bonded to bone. So this is where they have coined the terminology bioglass. Okay. So because there is a bioactivity, this bioglass can be bonded to bone within 24 hours, and bioglass can be bonded to synthetic tissue within 8 hours. So in fact, there is another history in 1969, as you can see. So here, Professor Yanas, in fact, he was again discussing with a doctor, clinical surgeon, and then after discussion, they realized, okay, body is made of more than 90% is collagen. How many types of collagen is there in our body? Anyone?
20.
1, 2, 3, 4, 5, 10, 20, 29 types collagens are there. But most abundant is type 1. Okay? Most abundant is type 1. So after discussion with the doctor, John Bart, so decided to go for collagen. So he realized, okay, because body is made of full of collagen, so if you put collagen, healing will happen much faster. Right? So therefore, so they made the collagen dressing placed on the wound bed side, supposed to heal faster, but it slowed down. So it has gone against the hypothesis. Right? So then what to do? So then they wanted to analyze what has happened in the wound bed. What they found that, quite interesting, that actually in a mother's womb, when baby has a wound, they heal without scarring. Right? So they found in the adult, when the wound was healed because of applying the collagen, healing was like a scar less healing. For the first time, they observed adult can have regeneration. That is the terminology they coined, regeneration in adult. So regeneration was there. People know regeneration, and regeneration terminology was coined also in philosophy a lot. Right? When you go to the euphoric state, then what you see, that okay, so now we have to come back to the original, then regeneration is supposed to happen. So that is why Yanas has realized by using collagen type 1, you can have regeneration. So let us see, 1969. Now you see that people did not know what is sterilization. 1920s, 25, did not know what is sterilization, what is antibiotics. When antibiotics came in, anyone? 1928, right? Antibiotic was developed in Oxford and another university in London, I found. Anyway, so and also people did not have the concept of sterilization. But that time they realized an egg can be easily fertilized. However, if the cell is broken, fertilization is difficult. Why? Because this membrane actually maintains the sterility. Similarly, in the springtime, you see egg has a hatching. So because spring water is sterilized water, right? This is very important. And also you see that these egg, when they hatch, they have a gel-like substance. These gel are actually a layer, barrier layer. This does not allow the virus or bacteria to invade into the creatures. So this is very important. So now if you look at the timeline, the stem cells was first discovered, I don't know when, but at least mesenchymal stem cells coined in 1991 by Arnold Kaplan, followed by 2024 or 24, US FDA approved the stem cell therapy. And fortunately, Government of India has approved the stem cell therapy in 2025. So I think I would say that all the tools are ready for all the young generation people to go ahead, jump on, do something. And interestingly, 2006, Jonas has discovered induced pluripotent stem cells. This was in the history the fastest Nobel Prize winning discovery, because just worked for 5 years, discovered in 2006, and got Nobel Prize in 2012. With this IPS, again I would say that I think the platform is set for work, cell-related therapies. Okay, so another path-breaking discovery. This is the last one, at least in my series of discovery story. So as you can see that there is a baby in USA was having more than 40% burn, and it was very difficult to save the life. Where the doctors decided to go for, again arbitrarily, let us go for culturing the cells on the tissue paper or filter paper. So they had grown the cells on the filter paper and they applied on the burn site. Let's see what happens. And they found that baby survived. That was the story of birth of Integra. Today, the lab Integra, the MIT lab is running full of profit money on the Integra. This is very important, remarkable. Okay, so with this, as you all know, this kind of stories, this is a very common story in India back in 2000 years back. Not this, there was a scar on the forehead of the human. Because in India, capital punishment was removing the nose and ear. So if you have to worship God without nose and ear, you cannot. It is not good. It is not acceptable. So therefore, what they will do? They will do the surgery. They will make cut like a nose, and they will remove, reverse it, and they will just stitch it. Reconstruction happens. So therefore, the person will have always the forehead having a scar. But this is a story in 2012 in China. What they have done? They have 3D printed a cartilage placed on the human forehead with a growth factor. Whole scaffold was converted into the cartilage, and therefore it can be used for reconstruction. Right? So with this, I think I would like to say, as you all know today, materials not only bioinert but also bioactive and resorbable, and of course bioactive, bioresorbable as well as bioinert. Okay? I have time or no?
Yes.
Time is there?
Yes.
5, 10, 20? 10 minutes? 15 minutes? Okay. So I think I joined back in 2007 in IIT Kharagpur with 4 primary ideas: digitization of the medical problem, ethically sourced material against sustainable materials, and customizing manufacturing solution, validation in vitro and in vivo. So the group was formed in Biomaterials and Tissue Engineering group. So of course we have done a lot of CT scan, MRI data, that reverse engineering. So when you have to choose the materials and the process, this is very, very important. I think just a couple of weeks back, maybe in the class also I discussed. So which material to choose? Should I go for sigma always for fancy material? Or should I use external material containing nano, like CNT, nanotubes, nanorods, graphene and all these? These are all fancy material. So you imagine that in your walk of the life there are many problems you are putting again and again, graphene, CNT and so many other materials. So your body becomes a chemical reservoir. So you are a living fossil where full of chemical is there. Anybody needs graphene? Okay, you dig up, then you get a graphene. Because it never degrades. This is very, very important. You can make a fancy material, you can use the fancy material, you can publish, doesn't matter. But what is the fate of material? That is why you need to go for a sustainable, degradable material. Okay, this is very important. So in fact, when you are choosing the process, I think 3D printing is everywhere now, but it is very, very important to understand. Is the 3D printing can do everything? So there are many processes. Depending on the situation, depending on the implant type, you have to choose different processes, right? So I'm not going to talk about the processes, but eventually we have two major processes. One is called top down and another is bottom up. Okay? So in top down, what you do, we have a heels cut into the elara, and in the bottom up, we have the bricks assembled into the building. Okay? So there are multiple processes with that. So now I'm just going again to the basics. Okay, and again, as I said, that Harvard has decided for absolutely bioinert material to put into the body. So failure analysis, people have realized that failure always happens at the interface between the bone and the implant, specifically for the load-bearing implants. So that is why Harvard decided to have a bioinert material, but macro porosity. What is macro porosity? Pore size should be at least 100 micron or above. What is the utility of macro porosity? When there is a macro porosity, always tissue will go into the pores. Therefore, and that tissue is a fibrous tissue. Because foreign body response, fibrous tissue will go into the pores. Therefore, there is interfacial bonding, or it's not interfacial bonding; it's a morphological fixation. But whereas Halbert said, no, bioactive material. So tissue will grow and it will be bonded to the material. So Halbert concept says growing into the pores, fibrous tissue, and hence said bioactive material. Therefore, cell and tissue will grow and bonded to the material. This is very important, right? So if you look at today's implant, this is a dental implant launched by Zimmer Biomet in 2014. This is a Brazilian lady, full 3D printed mandible coated with hydroxyapatite, and also today's implant, as you see here. So now all the implants, if you look at, they are not only rely on Halbert concept but also rely on Haynes concept. It is a combination of both the approach. In fact, I will tell you, in 2005, when Haynes was retiring, so my supervisor told me, let's go to the Haynes meeting in Imperial College. So we were there, Halbert and Haynes both are fighting. That day also they are fighting who is better, Haynes or Halbert. But if you look at the implants, you need to have both the concept. This is very, very important, because you need to have a macroporosity so that tissue can grow into the pores, but also you should have bioactivity so that tissue not only will grow but also be bonded. So look at the Zimmer, what they are coining is osteoincorporation. So not only ingrowth but also ongrowth. Why? Because at the interface, higher volume of material and higher volume of porosity will cross talk. Therefore, load transfer is not a point contact load transfer. It is a high volume of material and high volume of tissue will cross talk. Therefore, stress shielding can be minimized, and therefore you can have better load transfer. So I think I'll just quickly run. Maybe 10 minutes is a lot. Okay. So now I quickly run into my talk. So you can see we are making dental implants. We are trying to make Halbert concept, and of course you can see dental bridges. And Haynes and Halbert concept, can we make macroporous? Can we bioactivate so that grow into the pores as well as grow on the into the bonded side? It can also be bonded. So that way, so we are making porous titanium. Again, bioactivity is there. You can see the bioactive porous titanium. So that it is not only tissue will be bonded, but also there is a bioactivity and as well as bacteriostatic activity. Because bacteria can be pierced by this kind of nanoneedle, therefore bacteria will not be able to grow. So with that, we have done quite a series of experiments, and you can see that there is untreated titanium and treated titanium. You can see treated titanium has better ingrowth. And also, of course, we have done some 3D printing. Everybody is doing, I have to do also. Anyway, I'm just joking. Don't worry. So this 3D printed titanium, when we did animal experiment, remarkable thing. So you can see that porous titanium, dense titanium. So green machining of titanium, I think I am not going to discuss all this fabrication thing, but this is very important. Look at when we are using 3D printed titanium, plus at the defect site. So you can see there is a very nice bone bonding. So I was discussing with my student is in now Glidewell, US company, dental company. I said, look at this. See, what is the gap? There is no growth. He was bioinert. His eyes were bioinert. Because you have to understand what I am saying. Look at this. There is a very nice growth. There is no growth. So it says that you need to have exact size and shape as per the defect. There is a gap, it will not cross talk. This is very, very important. Anyway, so I wanted to do a lot of things on this, but they have to hurriedly submit the thesis and we have to go. So obviously I cannot say much. So with that, I think we are doing CT scanning and customized implant like this. This is a zygomatic orbital implant. Similarly, you can see a lot of porous titanium implants, and these titanium implants were made into dense porous material. And also I have done some kind of clinical trial. I think I met with this lady 2023, I guess. Yeah, 23 January. So I remember she is a Delhi worker, and she said she had a problem in the brain, then there was a surgery. Then after surgery I met her, then she said, can I work like before? Because I carry heavy load. So this kind of patients, actually they keep actually empty. So you can see from the visual that there is a depression because there is an empty defect. So that is why we put the Barber defects. We have done about 20 patients. Only 1% died because of cancer, not because of failure. And when we are doing follow-up studies, it's difficult to find because everybody had a good surgery and good treatment. I don't know, maybe good, but at least they did not have any kind of failure. That and there was no issue with the titanium related implant. So you can see here we had some follow-up study we have done after 1 year and 2 years. So that we are writing a paper on that. So similarly, so a lot of people are working with the Excel for making powder synthesis. I don't say about it. So I said instead of making that, let us go into directly make a cocktail and then print like a scaffold. So with that we are able to print and we are able to put in an animal model and you could see there is a healing. So similarly, so we are had some project in ICMR, we are trying to make the phalanges, and also we are combining electrospinning and 3D printing. We are able to make a lot of hierarchical fibers, and these fibers are fibrous, and you can see very good activities. And this is very interesting. Maybe 3 minutes. So there is a cornea, you can see. So cornea sometimes have a scar. Retina is healthy, but cornea has a scar because of that light is not able to pass through. So in that situation, can we do something? I think Dr. Falguni Bhoti happens to be my first PhD student. They are working on 3D printing. I thought I should not compete with my student. I'm just joking. Okay, so instead we can try something else. In fact, it is not my idea. Dr. Rai Chaudhary, who came to our institute in this same room, there was a debate between the MMST students and ours, and he was quite interested to work with, and we started working on this. Just this happened in this room. So then with that, he decided to go for cornea-like implant. So we made this cornea-like implant with the bioactivity of the surface so that can have better tissue integration. Similarly, we do a lot of work on silk-based cervical stain and also collagen-based scaffolds for 3D printing. So I'll run, okay? So you can see here, so we do electrospinning. So sometimes while we are doing electrospinning, one of my students came to me and said 5 minutes mixing, we get floppy mass. If you do 12 hours mixing, we get fine membrane. She said, this is emulsion, this is solution. What to do? I said, okay, let's go see whether it is emulsion or solution, do microscopy. When we did microscopy, see that emulsion, both are emulsion, only one case, bigger droplet, finer droplet, bigger droplet, so even floppy mass because of larger diameter. So we thought, quite interesting. So it's like a skin-like structure. This is basal membrane, this is a connective tissue. So can we do electrospinning on top of this so that we can see the keratinocyte on the top and bottom fibroblast. With that, you can see we are able to make lab-derived synthetic skin graft. Of course, we have done a lot of animal studies. Okay. So now I think I should talk about bioprotein iodination on nanodot. I think Dr. Mukherjee is here, Sai Mukherjee. Okay. So this work Again, I think one of my students, Dr. Bodhisattva Das, did some work on carbon dot, and I had a dream. We will load iodine a lot into the carbon dot. So I talked to him, can you do that? We started doing, and we could see remarkable result. We have done a lot of studies with the Tata Medical Center using the antibiotic resistant bacteria, and we could see that we are able to kill the bacteria. And of course we have used the infected wound model, and you could see the infected wound model also this carbon dot can kill. Okay, so these carbon dots were, again, the students were playing. Then we said, can you load the carbon dot into the nanofiber? So then you can see nanofiber are fluorescing actually. So now these nanofibers can be put into the wound dressing material. If you do follow-up study, only the fluorescent follow-up study, you don't need to touch the wound bed. So you can see what is the fate of the fibers inside the skin. This was remarkable. And not only that, these carbon dots can be taken up by the cells and can be fluorescing. So therefore, we can do follow-up. So usually the dye can follow up to 4 generations. We have done up to 8 generations and without any toxicity. This is very important. But we have not done zero toxicity time anyway. Okay. So interestingly, this DHF, whatever we say, carbon iodine loading, this DHF can be used for cross-linking to any of the NH2 containing polymers, any of the proteins, silk, and so on and so forth. And then while I am cross-linking, we can also just apply the iodine. So therefore, we can load the iodine, and this kind of polymers, if you see under X-ray, radiopacity is there. So, so this can be used for multiple applications. One of the applications could be where gauze is used instead of lead-based tagging, because sometimes by mistake gauze may be there inside the body. So that is why they use the lead-based tagging, so that by X-ray they can track the gauze. So instead, we can use the chitin-based material, silk-based material, or collagen-based material, or any other material, and it can become radiopacifier. So therefore, you can do X-ray and you can check whether it is by mistake it is there or not. Okay, so collagen type 2, you can see. Okay. So based on all the work, I think we formed a startup back in 2012, still struggling, but now we are able to sell quite a bit of collagen type 1, type 2, gelatin also, and the startup got best startup award in 2022 in IIT. And so we are using fish waste, many other waste material to make collagen type 1, type 2 and dressing. So we have a lot of clinical trial project going on. In fact, this startup and other institutes, 5 institutes together, we are making samples for clinical trial, and so we have such products. We have done some application of collagen into the human application in National Burn Center, Mumbai. You can see the where the collagen was applied. You can see scarless healing is there. Similarly, there is diabetic ulcer where actually applied and with this, I would like to say that we have explored a lot of things, but unless you go to the market, unless you sell it, has no meaning. So we are trying the next journey, and all of you, let us pray to me or to my startup so that it will have a success. Okay, thank you very much. So the students, lot of students, I think 20 or 40 students, I think at least the work I'm showing, and also the funding agencies and my collaborators, Dr. Kaji Zahiruddin, Dr. Punit Faljare, Dr. Izat, and Dr. Savarpal and Dr. Panda and many others, my supervisors. Okay, thank you very much. This more time I have taken,